A connectome is a comprehensive map of neural connections in the brain, and may be thought of as its wiring diagram. More broadly, a connectome would include the mapping of all neural connections within an organism's nervous system. The production and study of connectomes, known as connectomics, may range in scale from a detailed map of the full set of neurons and synapses within part or all of the nervous system of an organism to a macro-scale description of the functional and structural connectivity between all cortical areas and subcortical structures. The term connectome is used primarily in scientific efforts to capture, map, and understand the organization of neural interactions within the brain. Research has successfully constructed the full connectome of one animal, the roundworm C. elegans, White et al., 1986, Varshney et al., 2011. Partial connectomes of a mouse retina and mouse primary visual cortex have also been successfully constructed. Bach et al.'s complete 12 TB data set is publicly available at the Open Connectome project. The ultimate goal of connectomics is to map the human brain. This effort is pursued by the Human Connectome Project, sponsored by the National Institutes of Health, whose focus is to build a network map of the human brain in healthy, living adults. Origin and usage of the term. In 2005, Dr. Olaf Sporns at Indiana University and Dr. Patrick Hagman at Lausanne University Hospital independently and simultaneously suggested the term connectome to refer to a map of the neural connections within the brain. This term was directly inspired by the ongoing effort to sequence the human genetic code to build a genome. Connectomics, Hagman, 2005, has been defined as the science concerned with assembling and analyzing connectome data sets. In their 2005 paper, The Human Connectome, A Structural Description of the Human Brain, Sporns et al. Wrote, "To understand the functioning of a network, one must know its elements and their interconnections." The purpose of this article is to discuss research strategies aimed at a comprehensive structural description of the network of elements and connections forming the human brain. We propose to call this dataset the Human Connectome, and we argue that it is fundamentally important in cognitive neuroscience and neuropsychology. The connectome will significantly increase our understanding of how functional brain states emerge from their underlying structural substrate, and will provide new mechanistic insights into how brain function is affected if this structural substrate is disrupted. In his 2005 PhD thesis, From Diffusion MRI to Brain Connectomics, Hagman wrote, "It is clear that, like the genome, which is much more than just a juxtaposition of genes, the set of all neuronal connections in the brain is much more than the sum of their individual components. The genome is an entity itself, as it is from the subtle gene interaction that emerges." In a similar manner, one could consider the brain connectome, set of all neuronal connections, as one single entity, thus emphasizing the fact that the huge brain neuronal communication capacity and computational power critically relies on this subtle and incredibly complex connectivity architecture. Pathways through cerebral white matter can be charted by histological dissection and staining, by degeneration methods, and by axonal tracing. Axonal tracing methods form the primary basis for the systematic charting of long-distance pathways into extensive, species-specific anatomical connection matrices between gray matter regions. Landmark studies have included the areas and connections of the visual cortex of the macaque, Fellman and Van Essen, 1991, and the thalamocortical system in the feline brain, Scannell et al., 1999. The development of neuroinformatics databases for anatomical connectivity allow for continual updating and refinement of such anatomical connection maps. The online macaque cortex connectivity tool COCO-MAC, Cotter, 2004, and the temporal lobe connectome of the rat are prominent examples of such a database. In the human brain, the significance of the connectome stems from the realization that the structure and function of the human brain are intricately linked, through multiple levels and modes of brain connectivity. There are strong natural constraints on which neurons or neural populations can interact, or how strong or direct their interactions are. Indeed, the foundation of human cognition lies in the pattern of dynamic interactions shaped by the connectome. However, structure-function relationships in the brain are unlikely to reduce to simple one-to-one mappings. In fact, the connectome can evidently support a great number of variable dynamic states, depending on current sensory inputs, global brain state, learning and development. Some changes in functional state may involve rapid changes of structural connectivity at the synaptic level, as has been elucidated by 2 photon imaging experiments showing the rapid appearance and disappearance of dendritic spines, Bonhoeffer and Yusti, 2002. Despite such complex and variable structure-function mappings, the connectome is an indispensable basis for the mechanistic interpretation of dynamic brain data, from single-cell recordings to functional neuroimaging. The term connectome was more recently popularized by Sebastian Seung's "I am my connectome" speech given at the 2010 TED conference, which discusses the high-level goals of mapping the human connectome, as well as ongoing efforts to build a three-dimensional neural map of brain tissue at the microscale. In 2012, Seung published the book Connectome: How the Brain's Wiring Makes Us Who We Are. At multiple scales, brain networks can be defined at different levels of scale, corresponding to levels of spatial resolution in brain imaging. Cotter, 2007, Sporns, 2010. These scales can be roughly categorized as microscale, mesoscale, and macroscale. Ultimately, it may be possible to join connectomic maps obtained at different scales into a single hierarchical map of the neural organization of a given species that ranges from single neurons to populations of neurons to larger systems like cortical areas. Given the methodological uncertainties involved in inferring connectivity from the primary experimental data, and given that there are likely to be large differences in the connectomes of different individuals, any unified map will likely rely on probabilistic representations of connectivity data. Sporns et al., 2005. Mapping the connectome at the microscale, micrometer resolution, means building a complete map of the neural systems, neuron by neuron. The challenge of doing this becomes obvious. The number of neurons comprising the brain easily ranges into the billions in more highly evolved organisms. The human cerebral cortex alone contains on the order of 1,010 neurons linked by 1,014 synaptic connections. By comparison, the number of base pairs in a human genome is 3 times 109. A few of the main challenges of building a human connectome at the microscale today include: 1. Data collection would take years given current technology. 2. Machine vision tools to annotate the data remain in their infancy, and are inadequate, and 3. Neither theory nor algorithms are readily available for the analysis of the resulting brain graphs. To address the data collection issues, several groups are building high-throughput serial electron microscopes. Kazthurai et al., 2009, Bach et al., 2011. To address the machine vision and image processing issues, the Open Connectome project is algosourcing, algorithm outsourcing, this hurdle. Finally, statistical graph theory is an emerging discipline which is developing sophisticated pattern recognition and inference tools to parse these brain graphs. Goldenberg et al., 2009. A mesoscale connectome corresponds to a spatial resolution of hundreds of micrometers. Rather than attempt to map each individual neuron, a connectome at the mesoscale would attempt to capture anatomically and/or functionally distinct neuronal populations, formed by local circuits, e.g. cortical columns, that link hundreds or thousands of individual neurons. This scale still presents a very ambitious technical challenge at this time and can only be probed on a small scale with invasive techniques or very high field MRI on a local scale. A connectome at the macro scale, millimeter resolution, attempts to capture large brain systems that can be parcellated into anatomically distinct modules, areas, parcels or nodes, each having a distinct pattern of connectivity. Connectomic databases at the mesoscale and macroscale may be significantly more compact than those at cellular resolution, but they require effective strategies for accurate anatomical or functional parcellation of the neural volume into network nodes. For complexities see, e.g., Wallace et al., 2004. Mapping at the cellular level. Current non-invasive imaging techniques cannot capture the brain's activity on a neuron-by-neuron level. Mapping the connectome at the cellular level in vertebrates currently requires post-mortem microscopic analysis of limited portions of brain tissue. Non-optical techniques that rely on high-throughput DNA sequencing have been proposed recently by Anthony Zador, CSHL. Traditional histological circuit mapping approaches rely on imaging and include light microscopic techniques for cell staining, injection of labeling agents for tract tracing, or chemical brain preservation, staining, and reconstruction of serially sectioned tissue blocks via electron microscopy, (EM). Each of these classical approaches has specific drawbacks when it comes to deployment for connectomics. The staining of single cells, e.g. with the Golgi stain, to trace cellular processes and connectivity suffers from the limited resolution of light microscopy as well as difficulties in capturing long-range projections. Tract tracing, often described as the gold standard of neuroanatomy for detecting long-range pathways across the brain, generally only allows the tracing of fairly large cell populations and single axonal pathways. M reconstruction was successfully used for the compilation of the C. elegans connectome, White et al., 1986. However, applications to larger tissue blocks of entire nervous systems have traditionally had difficulty with projections that span longer distances. Recent advances in mapping neural connectivity at the cellular level offer significant new hope for overcoming the limitations of classical techniques and for compiling cellular connectome data sets. Livet et al., 2007, Lichtman et al., 2008. Using Brainbow, a combinatorial color labeling method based on the stochastic expression of several fluorescent proteins, Lichtman and colleagues were able to mark individual neurons with one of over 100 distinct colors. The labeling of individual neurons with a distinguishable hue then allows the tracing and reconstruction of their cellular structure, including long processes within a block of tissue. In March 2011, the journal Nature published a pair of articles on microconnectomies, Bach et al. And Brigman et al. In both articles, the authors first characterized the functional properties of a small subset of cells, and then manually traced a subset of the processes emanating from those cells to obtain a partial subgraph. In alignment with the principles of open science, the authors of Bach et al., 2011, have released their data for public access. The full resolution 1/2 TB dataset from Bach et al. Is available at the Open Connectome project. In 2012, a citizen science project called iWire began attempting to crowdsource the mapping of the connectome through an interactive game. Independently, important topologies of functional interactions among several hundred cells are also gradually going to be declared. Shimano and Beggs, 2014. Scaling up ultrastructural circuit mapping to the whole mouse brain is currently underway. Mikula, 2012. An alternative approach to mapping connectivity was recently proposed by Zador and colleagues, Zador et al., 2012. Zador's technique, called BOIN, barcoding of individual neuronal connections, uses high-throughput sequencing to map neural circuits. Briefly, the approach consists of 1. labeling each neuron with a unique DNA barcode. 2. Transferring barcodes between synaptically coupled neurons, for example using PRV, and 3. Fusion of barcodes to represent a synaptic pair. This approach has the potential to be cheap, fast, and extremely high throughput. Mapping at the macro scale. Established methods of brain research, such as axonal tracing, provided early avenues for building connectome data sets. However, more recent advances in living subjects has been made by the use of non-invasive imaging technologies such as diffusion magnetic resonance imaging and functional magnetic resonance imaging, fMRI. The first, when combined with tractography, allows reconstruction of the major fiber bundles in the brain. The second allows the researcher to capture the brain's network activity, either at rest or while performing directed tasks, enabling the identification of structurally and anatomically distinct areas of the brain that are functionally connected. Notably, the goal of the Human Connectome project, led by the WUMIN Consortium, is to build a structural and functional map of the healthy human brain at the macro scale, using a combination of multiple imaging technologies and resolutions. Recent advances in connectivity mapping. Over the past few years, several investigators have attempted to map the large-scale structural architecture of the human cerebral cortex. One attempt exploited cross-correlations in cortical thickness or volume across individuals, HEETAL, 2007. Such gray matter thickness correlations have been postulated as indicators for the presence of structural connections. A drawback of the approach is that it provides highly indirect information about cortical connection patterns and requires data from large numbers of individuals to derive a single connection data set across a subject group. Other investigators have attempted to build whole brain connection matrices from diffusion imaging data. Primary challenge for macro-scale connectomics: determining parcellations of the brain. The initial explorations in macro-scale human connectomics were done using either equally sized regions or anatomical regions with unclear relationship to the underlying functional organization of the brain, e.g. gyral and sulcal-based regions. While much can be learned from these approaches, it is highly desirable to parcellate the brain into functionally distinct parcels, brain regions with distinct architectonics, connectivity, function, and/or topography. Fellman and Van Essen, 1991. Accurate parcellation allows each node in the macro-scale connectome to be more informative by associating it with a distinct connectivity pattern and functional profile. Parcellation of localized areas of cortex have been accomplished using diffusion tractography, Beckman et al. 2009, and functional connectivity, Nelson et al. 2010. To non-invasively measure connectivity patterns and define cortical areas based on distinct connectivity patterns. Such analyses may best be done on a whole brain scale and by integrating non-invasive modalities. Accurate whole brain parcellation may lead to more accurate macro-scale connectomes for the normal brain, which can then be compared to disease states. Mapping functional connectivity to complement anatomical connectivity. Using functional MRI, fMRI, in the resting state and during tasks, functions of the connectome circuits are being studied. Just as detailed road maps of the Earth's surface do not tell us much about the kind of vehicles that travel those roads or what cargo they are hauling, to understand how neural structures result in specific functional behaviors such as consciousness, it is necessary to build theories that relate functions to anatomical connectivity. However, the bond between structural and functional connectivity is not straightforward. Computational models of whole brain network dynamics are valuable tools to investigate the role of the anatomical network in shaping functional connectivity. In particular, computational models can be used to predict the dynamic effect of lesions in the connectome. As a network or graph, the connectome can be studied as a network by means of network science and graph theory. In case of a microscale connectome, The nodes of this network, or graph, are the neurons, and the edges correspond to the synapses between those neurons. For the macroscale connectome, the nodes correspond to the ROIS, regions of interest, while the edges of the graph are derived from the axons interconnecting those areas. Thus connectomes are sometimes referred to as brain graphs, as they are indeed graphs in a mathematical sense which describe the connections in the brain, or, in a broader sense, the whole nervous system. One group of researchers, Auditory Medina et al., 2008, has constructed connectome data sets using diffusion tensor imaging, DTI, followed by the derivation of average connection probabilities between 70 to 90 cortical and basal brain gray matter areas. All networks were found to have small world attributes and broad-scale degree distributions. An analysis of betweenness centrality in these networks demonstrated high centrality for the precuneus, the insula, the superior parietal and the superior frontal cortex. Another group, Gong et al. 2008, has applied DTI to map a network of anatomical connections between 78 cortical regions. This study also identified several hub regions in the human brain, including the precuneus and the superior frontal gyrus. Hagman et al. 2007, constructed a connection matrix from fiber densities measured between homogeneously distributed and equal-sized regions of interest, ROIS, numbering between 500 and 4,000. A quantitative analysis of connection matrices obtained for approximately 1,000 ROIS and approximately 50,000 fiber pathways from 2 subjects demonstrated an exponential, 1-scale, degree distribution as well as robust small world attributes for the network. The data sets were derived from diffusion spectrum imaging, DSI, Weidin, 2005, a variant of diffusion-weighted imaging that is sensitive to intravoxel heterogeneities in diffusion directions caused by crossing fiber tracks and thus allows more accurate mapping of axonal trajectories than other diffusion imaging approaches, Weidin, 2008. The combination of whole-head DSI datasets acquired and processed according to the approach developed by Hagman et al., 2007, with the graph analysis tools conceived initially for animal tracing studies, Sporns, 2006, Sporns, 2007, allow a detailed study of the network structure of human cortical connectivity, Hagman et al., 2008. The human brain network was characterized using a broad array of network analysis methods including core decomposition, modularity analysis, hub classification, and centrality. Hagman et al. Presented evidence for the existence of a structural core of highly and mutually interconnected brain regions, located primarily in posterior medial and parietal cortex. The core comprises portions of the posterior cingulate cortex, the precuneus, the cuneus, the paracentral lobule, the isthmus of the cingulate, the banks of the superior temporal sulcus, and the inferior and superior parietal cortex, all located in both cerebral hemispheres. A subfield of connectomics deals with the comparison of the brain graphs of multiple subjects. It is possible to build a consensus graph such the Budapest reference connectome by allowing only edges that are present in at least k connectomes, for a selectable k parameter. The Budapest reference connectome has led the researchers to the discovery of the consensus connectome dynamics of the human brain graphs. The edges appeared in all of the brain graphs form a connected subgraph around the brainstem. By allowing gradually less frequent edges, this core subgraph grows continuously, as a shrub. The growth dynamics may reflect the individual brain development and provide an opportunity to direct some edges of the human consensus brain graph. The possible causes of the difference between individual connectomes were also investigated. It has been found that the macroscale connectomes of women contain significantly more edges than those of men, and a larger portion of the edges in the connectomes of women run between the 2 hemispheres. In addition, connectomes generally exhibit a small world character, with overall cortical connectivity decreasing with age. The aim of the as of 2015 ongoing HCP Lifespan Pilot Project is to identify connectome differences between 6 age groups, 4-6, 8-9, 14-15, 25-35, 45-55, 65-75. More recently, connectograms have been used to visualize full brain data by placing cortical areas around a circle, organized by lobe. Inner circles then depict cortical metrics on a color scale. White matter fiber connections in DTI data are then drawn between these cortical regions and weighted by fractional anisotropy and strength of the connection. Such graphs have even been used to analyze the damage done to the famous traumatic brain injury patient Phineas Gage. Statistical graph theory is an emerging discipline which is developing sophisticated pattern recognition and inference tools to parse these brain graphs. Goldenberg et al., 2009.
Please subscribe and thanks for watching.